But next up is our keynote lecture by Professor Ulrich Schurr, who is going to talk about quantitative approaches to improvements of crop and agricultural systems for a changing climate. Please, Professor Schurr.
Thank you very much, and welcome, distinguished guests. Thank you very much for the opportunity to talk to you today about quantitative approaches to improve crop and agricultural systems in a changing climate, which is definitely a very important topic, which we can also move on in the future together with the Vulcani Center. I don't think I have to go into extremely details with the challenges of agriculture in the 21st century, because many of the speakers before have already mentioned many of them. Therefore, I just want to shortly summarize the great challenges which we have ahead of us. On the one hand, we have to look at the environment. We see more and more weather extremes and climate extremes which are affecting crops. We have the water use and the contamination of water bodies, which are very important. Agriculture is a very important participant in. And we see a strong decline in biodiversity, which also is affecting agriculture and which agriculture is also a reason for. At the same time, we see a very much increasing demand in food and in non-food demands. At the same time, for both, for food and non-food demand, we want to have increased quality and increased diversity. And not to forget food waste and the loss of food which we have after the production has taken place. All of these are topics which are obviously very good interactions possible together with Israel and with the Vulcani Center. All of them, however, ask for an integrated systematic approach, because there is no single solution which is sufficient, but we need to integrate and have significant impact. We need to have integrated land use management, and we have to optimize the entire food, feed, non-food system together with the water energy agri-nexus. And this is asking for a circular agri and bioeconomy as a central element in what we use, say, in the European level on a Green Deal. So these are the challenges which we have ahead of us. But we have to talk about solutions, about options which we have. And obviously, the traditional activities towards breeding and crop management are towards yield and quality to improve the potential and actual yield and quality of crops. At the same time, we have to work on the inputs. We have to reduce fertilizers and chemicals and water input into the agricultural systems through management and through different measures which we can build on. For all these, we need to have quantitative data, quantitative and innovative data and solutions which are delivering into breeding and management. And there are mainly three different areas where we use them. One is to understand the systems, to understand the crops and to understand the agricultural systems. The second, to design them in a new and proper way. And last but not least, to manage the entire systems in order to achieve, on the one hand, yield and on quality, and on the other hand, to reduce the impact on the environment. And obviously, the task which is given to me today to talk to you in the keynote about quantitative data, I can only scratch the surface of options which are there. And therefore, I have to give you some examples. And the first example on the understanding where we see very important improvements in the past is about crop functioning under dynamic conditions of global change. And the example which I want to use here is root performance, which is very important because the root is the organ of the plant which is taking up water and nutrients and therefore determines a lot of the resource use efficiency. We have, in order to achieve these improvements, we have to, on the one hand, develop and apply unique technology. And both Germany and Israel are have very great history in this. We then have to quantify and model the crop performance in specific contexts, and I'll give you some examples on this later on. And secondly, then continue towards testing genotypes at future climates, in stressful climates, and then also putting them into genetic markers and idiotypes. And then we have to pass over these activities and these results to breeding programs. In Germany, this is often breeding companies, but also breeding projects in general. So to give you an example on the technology, we have developed in the past unique tomographic technologies for root analysis in situ. The root is the part of the plant which is in the soil, therefore we can't see it. And therefore, we use two technologies and further improved technologies, which you usually have experience in medicine: the magnetic resonance imaging and positron emission tomography. These two technologies give us the opportunity to study the root in situ, which means in the soil, with respect to their structure, as well as with their function with respect to carbon transport. The combination of both gives us a 3D trait co-registration, which allows to generate new opportunities for breeders to develop new traits. This results in things like this, which you see here. This is a time frame of roughly 2 hours, where we can see how carbon actually flows into root systems of the same specimen at 3 different days during the development: 6 days, 13 days, and 20 days. So this non-invasive technology for the first time gives us the opportunity to learn what actually happens below ground, which is extremely important for the response of plants to the environment and how the plants take up nutrients and water from the environment. We then have to test and understand how systems work in the field. There also we have specific facilities which we use quite nicely at the moment for testing many different genotypes, because many different genotypes are needed for breeding. And we are putting this in the context of a CO2 concentration in the field. So we mimic the future CO2 concentration in the field using our breed phase facility. This is also linked to a state-of-the-art field phenotyping devices, which you can see here as well. This is a kind of 3D scanner, which gives us very detailed information about yield, photosynthetic traits, water relations, structural traits, but also about insects and pests at future CO2 concentrations in the field in Germany. We can then put this into the next steps by using agrorobotics. And we have recently formed, with the funding of the German Science Foundation, an excellence cluster in agrorobotics, where we combine our quantification on phenotypes and environment from the level of single plants up to the level of the satellites and global, with agrorobotics, sensing, and machine learning to get quantitative data out of this data. But this is not enough. We have to combine it with analysis at multi-scales about economic, ecological, and social impact of these new technologies in order to be able to build this into a bigger context and to generate impact in the real world. We built this now into a whole digital farming landscape, also in collaboration with colleagues from the BML, in collaboration with 5G technologies, artificial intelligence, and data sciences. So many of these new technologies have direct implications for the activities which we do in agriculture. In the next step, we go beyond the German level, and I want to shortly mention here the emphasis S3 project which we have, where at the moment we are building a pan-European plant phenotyping research infrastructure. This addresses controlled environments, intensive and lean field environments, modeling and data and computational services which are provided with this research infrastructure. We have in the decision body at the moment 12 different governments from Europe, and we are also happy that we have additional countries, including Israel and the Volcani Center, associated to this European research infrastructure. We then have to design how things are developing. And an important topic for that, obviously, is to use biodiversity for climate adaptation, because many of the natural plants or the wild plants have opportunities to work on different climates. And therefore, we use bioinformatics to implement new crop traits. We identify and use heritable properties of wild species, for example, tomatoes, and this has also very strong interaction there with Israel. We then do sequencing, and we did the first nanopore sequencing with new technology for tomatoes in plants, and combine this with phenotyping technologies. And by doing this, we can generate a plant genetic analysis framework, which allows us to improve crop resistance in future climates. This is design opportunities which we have to use also in the future. Again, here we have an excellence cluster, which is also following along that line of design, where we use biodiversity to crop traits for crop development, primary production, metabolism, but also integrate a microbiome in the virome of the soil, which is a very important part of the overall performance in the environment. Combining with synthetic biology and theoretical biology, this gives new opportunities to design crops from scratch to the future developments. We also have to think about non-food applications, and therefore we utilize renewable carbon sources for value-added products. We can use diverse feedstocks and residues, and we have to use them in order to make more efficient renewable products. We've built modular, hybrid, and biotechnology processes from biotransformation, enzyme cascades, and chemical synthesis, and we will also have some examples on this later on, in order to produce low footprint and environmentally friendly products. This is also a very important part of our bioeconomy efforts. And last but not least, we also have to manage the entire agrogeosystems. We have to quantify the environmental impact, the control and the feedback loops which are there in the environment in order to run the biogeosystems in a proper way. We have to develop and integrate multi-scale observation simulation platforms where we are already extremely active in doing this in the field, and to fundamentally understand biochemical processes across scales from small field parts to the globe. This will give us the opportunity to target ecosystem functions to reduce anthropogenic impact and to design sustainable management of crop protection systems in collaboration with our colleagues in Israel and in Germany. Again, here we have S3 project, which is running a long-term ecosystem research, the LTER, where the mission is to track and understand the effects of change on socioeconomic systems, including agriculture, and the feedback to environment and society. This will also provide recommendations and support for solving current and future environmental problems by networking, research and development, and infrastructure. Ladies and gentlemen, I hope that I could use the last 15 minutes to give you a short overview of what kind of quantitative approaches are there to improve crops and agricultural systems in the changing climate, and how many links there are to the work between here, what we do in Germany with the colleagues in Israel, specifically with the Vulcani Center. And I'm looking forward for the implementation of our MOU in the future and close collaboration. Thanks very much.
Thank you so much, Professor Schurr. Indeed, really exciting.